Hi, my name is Junhyuk Jang from Phillips Exeter Academy. Today I'll be talking about how microgravity contributes to prolonged stress by impairing ACTH regulation as it undermines the gene expressions of several ubiquitination-related genes. The 2020s is a decade where human spaceflight is a goal pursued by both governmental organizations and private industries, and as such, many astronauts are required to do research in unfamiliar domains like space. However, this is a very burdened task for the physiology of the astronauts, and it causes many dysfunctions such as mental stress. A primordially common stress response mechanism among all vertebrates is the hypothalamic pituitary adrenal axis, known as the HPA axis. Primarily, when stress stimuli is inputted, the BNSD, the bed nucleus of the stria terminalis, sends corticotrophin factors, known as CRF, to the hypothalamus. The paraventricular nucleus of the hypothalamus then relays CRF to the anterior pituitary. There, by means of a CAMP pathway, the precursor POMC is converted into the hormone ACTH. This hormone then travels to the adrenal cortex, and it binds there to release cortisol. Now, to prevent excess cortisol from being happened, since the cortisol is a hormone that activates the fight or flight response, there is negative feedback to control excess hormone interactions from happening. Ubiquitination is a three-step process that involves the destruction of hormones and other unnecessary molecules in the body. Three enzymes are responsible for this: E1 for ubiquitin activation, E2 for ubiquitin conjugation, and E3 for ubiquitin protein ligation. After the target molecule has been attacked by ubiquitination, it is destroyed by a proteasome. In the HPA axis, in the anterior pituitary, the POMC is converted into ACTH. To prevent excess conversion, as dictated by negative feedback, POMC is ubiquitinated. The GSE12312 dataset is a collection of hind limb unloaded mice and see how their brain gene expression changes under the condition. After analyzing the data, I found that the genes UBE1C, UBE2I, UBE3B, each responsible for the steps in ubiquitination, were all downregulated. Therefore, it impairs POMC degradation, and therefore there will be excess ACTH synthesis, which means there will be a perpetual cortisol release. Therefore, since cortisol or the fight or flight response is perpetuated, there is a heightened stress risk. Because microgravity prolongs cortisol release and therefore the fight or flight response, astronauts are extremely vulnerable to stress. Therefore, wearables should constantly monitor how astronauts' mental state levels are and to manage stress when appropriate.